Hello, everybody here in the audience and out on Facebook Live, and welcome to the first edition of the Sci-Café for the 2018-2019 academic year. Thank you for joining us. The topic today is Toolbox biology, which I think is a catchy way to describe what we're going to be discussing. Just to peel things back a little bit and explain that at the beginning, I'm curious how many of you in the audience are biologists by training? Okay, so for those who can't see on Facebook Live, it's about a third of the audience, which is good, because I think some of today's discussion is going to be for the biologists in the room to learn more about some of the techniques that are being used here at KAUST. But I think a lot of today's discussion, for a chemist like me, biology can sometimes be a black box. It's especially modern biology with the genetics, the proteomics, the CRISPR-Cas9, the stem cells, the differentiation, all of these things that happen in modern biology. It can be hard to understand what's really happening at the cutting edge of research. So the group that we have here is going to help explain that as we go through the discussion today. So just to remind you of the format, I'm going to ask a few questions. I'm going to set the stage so you can figure out who these three experts are and what type of research they do. And in about 15 minutes, 20 minutes, I'm going to open it to you for questions. So please be ready to answer the call when we get to that point. So the way I'd like to start is I always like for the faculty to introduce themselves and tell a little bit about the field that they work in and what particularly excited them or brought them to work in that field. So Jürgen, maybe you want to start?
Sure. Thank you very much for inviting me to be here tonight. My name is Jürgen Kosel. I'm Associate Professor of Electrical Engineering, and I came to KAUST in 2009 as a founding faculty member, and I came from the automotive industry. And what I do in KAUST is working on micro and nano devices, basically small devices, and I use them for different applications, but our main focus is biomedical applications. And I'm just going to mention a few examples so that you get an idea. I work on surgical instruments that help the surgeon, for example, to reduce the X-ray exposure of patients. So I make smart devices that have sensors so that the surgeon can see, the heading or the position of the device instead of using X-ray. Or I make micro needles to deliver drug into arteries, and then you can treat arteriosclerosis long before you would use a stent or any of the common methods. But probably what is more interesting to today's talk is my work on magnetic nanowires. And I use them as nanorobots for biomedicine. And the nanowire is actually something very simple. It's like a stick or rod, and it's magnetic. And because it's magnetic, I can remote control it. And that gives me a lot of different capabilities to work with the nanowire inside of a body or inside of a cell. So for example, I can use the nanowire as a drill. And when you have a drill, you can kill cancer cells by drilling into them. Or I can use them to heat them up with a laser or a magnet, and again, that kills cancer cells. I can also use them as a truck loaded with drug that kills cancer cells. Or I can put them into cells, cancer cells, stem cells, or whatever, and then I can drive the cells inside of the body and thereby tell them where they go. And at the same time, I can visualize them outside with magnetic sensing systems like magnetic resonance imaging that probably most of you are familiar with. And the final aspect I'm working on right now with these nanowires, I can use the stick to trigger stem cells to become bone cells. So basically, these nanorobots, they have a lot of interesting features individually, but I can also combine them to make them even more capable, multifunctional properties. And I guess that's probably a good introduction.
So if we were going to maybe oversimplify it in terms of the tool that's most important in your toolbox biology, that's kind of the ability to control these nanomaterials and thereby control what they can target, what they can do in the body. Is that accurate?
That is right, yes.
Okay, good.
And this control is remotely, so I don't need any wire or cable to connect, but I can do this from outside. And magnetic fields are harmless to the body, can go through the body, and can thereby steer wires or detect wires and so on.
Very good. So this is nicely connected to actually a collaborator of Jürgen. So Jasmeen, maybe you want to introduce what you work on?
Hi, I'm Jasmeen Merzaban, and I'm assistant professor in the Biological and Environmental Sciences and Engineering division. I've called KAUST my home for the past 9 years. And my lab has many focuses, but one of the main focuses is adhesion systems, and how adhesion systems, we try to understand how these adhesion systems in cells, whether those are stem cells or cancer cells or immune cells, how these adhesion systems can guide these cells throughout our body and direct them to different tissues, okay? So we really, cells are moving at very high speeds in our bloodstream, relatively high speeds. You can think of it as the cells moving in our blood like a car is riding down a highway. And in order for it to get out of the bloodstream and into a particular organ or a tissue, they need to slow down. And these adhesive systems on the cell that connect the cell to its environment, and in this case, the blood vessels and the endothelial cells that line those blood vessels, those adhesive systems help to slow down the cell and also attract it to these different tissues. Now, we want to try to identify and to understand, but also to manipulate these cells so that we can either inhibit their movement around the body, in the case of cancer cells and metastasis, or to promote their ability to move to specific tissues, like in the case of therapeutic stem cells. So really, what we do in the lab is try to understand these adhesive systems and see if we can modulate them so that we can cure or treat diseases.
Right. So in terms of, in the body, all of those cells and all of that movement is a very complicated system.
Yes.
So what sort of tools do you use to then focus in on specifically that adhesion phenomenon?
Okay. So we use a lot of biochemical and cell biology-based tools, but one of the strongest tools that we have is actually imaging. So using a microscope and actually visualizing, mimicking what we would see in the bloodstream if we could actually see what's happening in the bloodstream. And so we can take endothelial cells, those cells that line our blood vessels, and put them in a petri dish, and then flow the particular cells of interest over top, and we can actually then see what's going on. If we remove some of these adhesive systems, then we can see, okay, what are the repercussions on the cell? We also try to understand what creates these adhesive structures. So we know that from immune cells, for instance, that inherently know how to get to an inflammatory site in our body. If we get sick, we know that our immune system is going to activate, and then those immune cells are going to go and fix that. And they inherently know which way to go because they have they have these enzymes that create these adhesive structures on their surface. Mm-hmm. And what we could learn from that is that we know that these enzymes are there, so now if we can isolate those enzymes and express them in high quantities, we can start to use them for our benefit and in treating, for instance, in directing the migration of stem cells. Yeah, we also use, so that's imaging, we also have collaborations with a lot of, with Professor Satoshi Habuchi, and he is a very talented imaging expert at KAUST, and he can actually do super resolution, real-time imaging of biological systems, okay? And so we can actually look at a confocal microscope and we can see these little round cells rolling or moving along the endothelium. But what he can do is give you even more resolution. And now we can actually image adhesive molecules on the cells themselves. So we can look really at the architecture of that cell, and we can see that it's more than just what we see, a round ball moving along. We can actually see that the cell is extending these arms out, and on those arms, there's adhesive molecules moving along, almost like a highway, an intrinsic highway on these adhesive arms. And those adhesion molecules are then making contacts, and these are dynamic contacts. Okay, so there's a lot of wonderful tools that we can use here at KAUST and also take advantage of collaborations. Yeah. And then, of course, we have a wonderful collaboration with Professor Kosel, and we are looking at nanowires and how we can target those nanowires to cancer cells. And we can talk about that a little bit later.
Yeah. So essentially, understanding the fundamentals of how these cells communicate and how they adhere to different surfaces, you then want to use that in different clinical applications is the end goal, correct?
Of course.
Good. Antonio, maybe you want to say a bit about your work?
Of course. So my name is Antonio Adamo. I'm an assistant professor in BESC division. I work mainly with human embryonic stem cells, so cells that derive from supranumerary in vitro fertilization derived embryos, but also with induced pluripotent stem cells. These cells, induced pluripotent stem cells, come from patients. They can be healthy donors, or they can be patients affected with a certain disease. And then we reprogram those samples to something similar to the embryonic stage, because we want to understand what are the molecular mechanisms that are broken or dysregulated in the tissues affected by a certain disease in those patients. So in order to do this, we apply several molecular techniques. Probably the one that you have been hearing about in the last few years is CRISPR-Cas9. Is a gene editing technique that allows us for the first time to modify the genome of a cell without introducing major rearrangement, over bypassing the need of using viruses, and we can basically modify the genome, but since we can do this, we can also basically fix whatever mutation, deletion, duplication is in this genome, and then apply these cellular tools that we derive to regenerative medicine applications. Of course, this is also a tool in which we can not only monitor the progression and the onset of a certain disease in a dish, but is also a tool that is extremely useful to perform drug screening assays that, as you can imagine, have an enormous impact in the pharma companies, and therefore there is also an enormous potential in terms of economic development.
So break it down a little bit more for the chemists in the room. So the tool you're essentially talking about is you take cells from a living person or from an animal source, whatever, living cells, and the tool you use is to essentially reprogram them backwards.
Exactly.
To revert them to a pluripotent state, and then that is now your tool. You have what do you call it, a disease in a dish?
Disease in a dish is
right?
perfect.
And then, so tell me about then, now you can test on that tool. That's then a platform for doing all different types of studies?
Okay, so I will give you an example. Perhaps it's easier. We work with diabetic patients. So we take cells from diabetic patients or patients that have a high risk of developing diabetes. We bring them back to the pluripotent stage, and then we differentiate in a dish these cells into glucose-sensitive cell types. So think about hepatocytes, cells from your liver, or adipocytes from your adipose tissue, or even more importantly, beta-pancreatic cells, that are those cells that respond to the glucose in your blood and release the insulin, and insulin is what is lacking in type 2 and type 1 diabetic patients. Okay, so we start from these embryonic stem cell or induced pluripotent stem cells if we talk of cells from patients. We differentiate them in vitro. This is just the first stage of differentiation. We apply three-dimensional growing techniques. Why three-dimensional? Because it's been demonstrated in the last decade that if you do it in 3D, you basically mimic the development of an embryo better than if you do it on a 2 dimensions. So in a dish attached to a plastic plate, and then we differentiate them into beta cells, for example, and we test them by challenging them with glucose, and we see whether the cells coming from a diabetic patient respond better, or as you can imagine, worse, compared to a healthy controls. Yeah. So that's what we do. Once we demonstrated we can mimic those diseases in a dish, then we use these cells to perform drug screening assay and to see whether we find a molecule that revert the dysregulated release of insulin in the diabetic patient-derived cells into a normal one.
Yeah. So basically fix the pluripotent cells to make, essentially try and fix the diabetes, yeah?
Exactly.
Very cool. So I'm going to preempt the audience questions, but be ready. I'm very curious. You know a little bit about the background and what they're working on, but maybe each of you can say a little bit about maybe clinical collaborations or where you're trying to take this in terms of an endpoint application. So maybe Jasmeen, you want to start?
Okay, so I think I'll talk about the stem cell aspect, and then maybe Jürgen can touch on what we would like to do with cancer cells. Okay, well, stem cells under the umbrella of stem cells, there's two major types of stem cells. There's the adult stem cells, and then there's the embryonic pluripotent stem cells, which Professor Adamo works on. So I work on adult stem cells, and adult stem cells are in all of us, okay? So they are stem cells that are found in different tissues and organs in our body. And the purpose of those cells is actually to generate or they're the mother of the specialized cells within that tissue. So for example, in the brain, a neural stem cell would give rise to all of the functioning cells within your brain. So it would be your astrocytes and your oligodendrocytes and your neurons. And they're there to replenish those cells when, after their lifetime is complete. So they have a finite lifetime, so they will eventually die. And these stem cells will replace those cells. Now imagine that you had, there's a neurodegenerative condition, or you had a stroke, or a heart attack, or you have a blood disease. In situations like this, these stem cells that are in those tissues are not able to replenish all of those cells that have passed away. So we need to do a transplant of stem cells of some sort. So either we take the stem cells and we grow them up in the lab, and we inject them or we put them into the organism, or we differentiate them like we do with Jürgen, down a specific lineage so that they would give rise to a specialized cell which we can then transplant into an organism. But the problem with stem cells is that because they stay in these tissues, inherently, they stay in that tissue. They don't need to move around the body, more or less. There are some examples where they do move around. But so they don't actually have these adhesive mechanisms on them. And so what we try to do is try to figure out what's missing. And one of those when we looked at these immune cells, we found out that there's these enzymes that are important in creating some of these adhesive structures on their surface. And these stem cells are lacking them. So when we isolated them and we grew up with a collaborator in Japan using a silkworm expression system, we can make a lot of these enzymes. And then we can add on what's like a sensor on the surface of that stem cell. And now it's able to migrate. It's able to find its way through the body into those distress zones areas where we need them to go, whether it's the brain, or whether it's the heart, or whether it's the bone marrow. And so this is one of the toolboxes that we use, is really to use expression systems where we can make enzymes or proteins in high amounts that can then be used for therapeutics.
Nice, very cool. You want to talk about the cancer side of that?
Yeah, or more generally, the clinical applicability of this. I think the important aspect is you develop something for biomedical applications. You need to work with biomedical people like clinics. And we collaborate with different people in that regard. We work with people in Jeddah, King Abdullah International Medical Research Center. There's a cardiologist who we work with. There is an oncologist who we work with. We have a pediatrist in South Africa who we work with to develop a device for infants. And we work with a group in Spain when it comes to using nanowires as magnetic resonance imaging contrast agent. And how relevant are nanowires generally, or nanomaterials for clinical applications, is probably best to talk about one material, that is iron oxide, that has been approved for clinical applications a long time ago in the form of balls, nanoballs, spherical, maybe 5 nanometer, 10 nanometer in diameter. And they were used as magnetic resonance imaging contrast agent on a routine basis, and were the first nanoparticle ever approved by FDA. And then, probably 15 years ago, people discovered that you can heat them up from outside and thereby destroy cancer cells. Now, since about 7 years, there is a company in Germany that provides these particles together with the equipment, and it's used in about 8 hospitals now to actually treat cancer and tumors. And they, of course, for now, they treat only the very severe cases, and they have an extremely high success where they can basically, statistically now, they can double the lifetime or life expectancy of the people who have certain types of cancer. So nanomaterials, especially iron oxide, are used since a long time in vitro, and they are used now for cancer treatment. And the materials we work with for bio applications are iron materials with an iron oxide shell or just pure iron oxide nanowires. So the materials have the material basics to go straight into clinical applications.
Very good. Maybe you want to expand a little bit on some of the, some of the clinical applications you're targeting?
So basically, right now, there are already phase 2 and phase 3 experimentation, phase trials for biocompatible devices. These biocompatible devices, you have to imagine them as capsules where cells that have been derived from embryonic stem cells and differentiated into beta cells have been embedded. This biocompatible device protects these cells from being attacked by the immune system, so you can transfer and transplant these devices under the skin, and they will basically act like if they were a pancreas for these patients. So when the glucose will go up in your bloodstream, these devices will let the cells that are embedded inside to release the insulin because they have porous on their membrane, but the immune system cells will not be able to attack the cells that are inside because they cannot get in. So this is what's going on right now. However, these biocompatible devices are needed because you use embryonic stem cells to derive these beta cells. So they are not recognized as self, and they would be attacked if you don't protect them with these devices. So what we are trying to do in the lab instead is to use cells that derive from the patients themselves, so that if you are able to differentiate them properly in beta cells, you can use them directly for regenerative medicine applications.
Very good. So hopefully that gives you an idea of the tools that are being used in some of the labs here at KAUST and some of the clinical applications. What questions do you have for these individuals about the science or the applications or any of the above? Everybody but Samir is allowed to ask a question. And this is not a football, this is the microphone.
Hello, thank you very much. My name is Bruno. I have 2 questions. One, what is the perspective on the lab meat or clean meat, like as a part of like a not vegetarian option, but just do you see any commercial future for the lab meat, which means the meat that is produced in the lab without by culture cells? That's one.
Okay.
And just a quick second one is, what is the bottleneck, or what are the challenges in stem cell research in terms of ethical background?
Yes.
What's still the limit there? What would you would like to happen regarding just the ethical use of stem cells? Thank you.
All right. So why don't we tackle the second one first, because I think you two can tackle that one, and then we'll go after lab meat.
I go with the second one?
Yeah.
So regarding the ethical aspects, I have to say that first of all, there has been a major advancement in 2006 when the Nobel Prize for Medicine, Shinya Yamanaka, described for the first time the technique of reprogramming. So before the 2006, in order to study development and regenerative medicine, we were using human embryonic stem cells. So we were destroying supernumerary donated embryos from IVF technology in order to study development. When Dr. Yamanaka described the reprogramming, we could use cells from any patient in the world. So we didn't have the issue of destroying an embryo. So this was a major change in the ethical aspects of working with embryo-like stem cells, okay? This is the first thing. But now I want to be a bit provocative and ask you to think about something. If we are able to do regenerative medicine for whatever tissue of your body, and we can also do regenerative medicine for your neurons, is this going to have an impact on your personality or on your behavior? And how long do you want to live? So there are definitely new challenges that need to be answered and addressed, but I would say that the major ethical concern of the work with embryonic stem cells have been overcome by the advent of induced pluripotent stem
Just to
cells.
challenge that a little bit, can you do everything with an induced pluripotent cell that you can do with a human embryonic stem cell?
Virtually, yes.
Virtually, yes.
Yes.
Fantastic. Did you have anything to add to that? Or?
Not really. I mean, adult stem cells, there isn't too much ethics, and actually for more than 5 decades, we have been doing bone marrow transplants to treat blood diseases. And bone marrow, the reason that it works is because it has a blood stem cell in it. So it resides there. So there's not too many ethical issues when it comes to adult stem cell work. It's mostly with the embryonic stem cells.
Unless Antonio's future comes to pass, then we have some ethical questions.
Exactly.
Maybe
This is particularly relevant if we talk about neurons, of course, because it affects personality and behavior. When it goes to hepatocytes for your liver or your pancreas, nobody or most of the population would never argue against those kind of regenerative medicine protocols, of course.
Yeah. Maybe you could say a little bit more, or do you guys have enough to answer the question about lab meat, kind of lab-created organs, I think is what you're talking about?
No, I'm talking about culture meat, so for eating, which is produced in the lab. That I know that there are some commercial burgers that are available for that, but they are quite expensive.
So I'm not really an expert of the field, so I will just give you my gut answer. First of all, I'm Italian, so I love food, good food. However, if this is a technology that is available and we will face, probably not in our generation, probably not in the next one, but you don't know what is going to happen in 100 years from now in terms of food availability, the idea of having an opportunity to have an option, I think we should explore it.
Cool. All right, what other questions are out there? You got to toss the football as best you can.
Okay.
Come on, you got it. Very nice. Go ahead.
Thank you very much for the talk. Name's Ali. And so I also have two questions. So one is kind of directly related to the ethical question. I specifically want to hear if you faced any kind of issues, especially being in Saudi Arabia and doing human stem cell research. Were there any issues trying to get that in here? Because I know in the US and Europe, they're always facing a lot of issues doing research with these type of cells.
Yeah.
The second question is kind of across. So you guys talked about biocompatibility and all of that. Did you guys also investigate in your research, I guess, if the iron spheres, for example, if they degrade or are they retrievable? Because can you just leave them in the human body after you use them, or is there some method at which they're disposed? Thank you.
So let me tackle the first one with a different hat. So I'm also in charge of research compliance here at KAUST. And so just to answer your question quickly, Saudi Arabia has a lot of the same regulations that exist in the US and elsewhere. They're kind of globally recognized standards. A lot of them come from the National Institute of Health and some of these best practices worldwide. So we essentially face the same regulations here that you would face anywhere, and Saudi Arabia has a nice network in place in order to make sure we can get the samples, that we're using it ethically. KAUST has all of the committees and the governance in place to make sure that we check that before the faculty start their work. They can probably give examples of how rigorous that process can be. For the second question, I was hoping somebody was going to ask this because I want to hear Jürgen's response on how biocompatible and how biodegradable these things are. So,
all right. Biocompatibility is actually quite a complex topic. Usually when you develop new nanomaterials, you do cytotoxicity assays or cell viability assays. We do that with Jasmeen's help. But you only get actually an understanding of whether cells cultured on a dish, whether they can deal with or handle the nanomaterials. But that doesn't mean that inside of the system, of course, of the biological organism, this is biocompatible. However, this iron oxide beads that I mentioned before, there have been a lot of studies done because they were used a lot in humans. And the first biggest challenge is you inject them in the bloodstream and the immune system recognizes them as something strange and wants to take it away, as Antonio described also before. So that's one big challenge that has been overcome by coating, which it's like it makes the particles stealthy and the immune system can't see it anymore. And that works really well. It even helps to keep them dispersed because they have some negative charge and repellent forces. So that was really good. So then they circulate in the bloodstream, but eventually they get filtered out. And it depends on the size of the particle what actually filters it out. Smaller particles are filtered by the kidney. and you dispose them when you go to the bathroom. Larger particles, they get trapped in the liver and spleen, and then you can see them over time, how they start disappearing because they get biodegraded. And the nice thing, I guess, about iron oxide is what you end up with is iron ions, and the body reuses them for hemoglobin or something that you need the ions for, and after a couple of months or a week, depending on the dose, you cannot see any remainders of these nanoparticles in your body anymore.
So let me ask a follow-up as the chemist again. So you do this work in a dish, like you said, and you're relying on historical studies that are in the human body, but how would you take some of your nanoneedles right now and start to transition them and find out, okay, I know they work in a dish, what's the next step?
So the next step is actually animal experiments, and we started animal experiments with our collaborators in Spain, but now we also have a very nice animal facility here in KAUST. You know especially well that it is operational. Thanks actually to you. We have started putting everything in place to inject these nanomaterials into the mice to see how they end up in cancer tissue, to see how we can actually guide stem cells, for example, to the bones, to the spine, and so on and so forth. So yeah, our next step is the animal experiments.
Very cool. What other questions are out there? Yeah, who's got the cube? Thank you.
Thank you very much for such a nice discussion. My question is Professor Kosel. You mentioned about magnetic nanowires and using that for treatment of the cancer cells. What do you think about what their side effects will be on the healthy cells? Because we have different technologies present now, like the gamma knife treatment. They say that it's very targeted, but ultimately it affects other body functions as well. So killing the cancer cells, how targeted will be using those magnetic nanowires?
I mean, this is a problem or a task and a challenge at the same time for all nanoparticles that you want to use inside of the body. Actually, for every drug. How does the drug know it's a cancer cell and not a healthy cell? And that is mainly the problem of chemotherapy. It goes everywhere, and everything that reproduces quickly or proliferates fast is being affected. Now, there is an effect that is specific to cancer cells, that they have a higher permeability, and nanoparticles go more into cancer cells than in healthy cells. And then there's also a retention effect that is more pronounced in cancer cells than in healthy cells, and that allows you to passively target. And this has been shown for these beads, again, these iron oxide beads. You inject them in the bloodstream, and then they accumulate inside of cancer cells passively. This is a very nice mechanism that works for a lot of cancers, apparently. But then, of course, there are these attempts to actively target also cells. This is, again, a strong collaboration with Jasmeen, where she's an expert in. And we functionalize the nanowires with antibodies that recognize cancer cells. And that is a common method that people who work with nanomedicine try to employ and utilize. And this is probably the right time when I hand over to Jasmeen.
Okay, so a little bit about cancer. Cancer is really smart. So it often uses similar molecules that normal cells have. The specific example that Jürgen is talking about is an adhesion molecule that is expressed on a lot of cells in our body, so normal cells and cancer cells. But there are variants of this molecule that are only expressed on cancer cells. So we have antibodies that are targeted to these variants so that we can then be more specific. And we've actually shown in other research in our lab that even if it is expressed on a normal cell, because of the level of expression, cancer cells tend to overexpress a lot of the adhesion molecules, or these adhesive systems, that they need to get to different places in the body. So they really have high levels. And the nanoparticles actually work by number. You have to have a certain concentration on the surface of the cells for it to work. So in that case, we can more readily target the cancer cell versus the normal cell.
What other questions are out there? Yes,
Okay.
sir.
I've got two questions. The first one is maybe off topic, but I wanted to ask it because I'm a little bit curious. You talk about you are working with nanomaterials to target cells and to develop new kind of therapies. I studied a little bit time ago that they were starting to use DNA to build like sensor and three-dimensional structure to build nanobots and stuff like that to actively target and deliver substances or enzymes, molecules into the cells, or to use it as a linker between cells, or to link non-organic particles with organic particles. Is it still used? It was only stuff that was discovered and then never applied, or because I don't know the situation right now. And second question is about CRISPR. And how do you actually use CRISPR-Cas9? Like, can you use it in situ, like by injecting it some kind of mixture of RNA and enzyme in situ? Do you have a local effect? Is it risky for the patient, and does it work?
Yeah. So let me break that down. So essentially what you're asking is DNA or other types of molecules. He's asking what the state of the art is in terms of, almost like nano robots, kind of where the field is headed. I mean, what sort of insight do you have there, Jürgen?
So this is the same field but different branch. This is the organic nanoparticle development versus what I'm working on is the inorganic one. The organic ones are an extremely active field too. They have the advantages and disadvantages, I would say, like inorganic ones. The major difference, or one major difference, is you have no active mechanism, say, to use a laser for heating it up or a magnet to guide them. They are used only as drug delivery. But that's the only modulus of treatment that you have. You cannot add anything other than the chemical treatment modality. And it's a problem to visualize them, which you cannot do with MRI or so too. So different branch, very exciting, very hot topic, lots of work is still going on.
And then he asked the same question I wanted to ask. So you're doing it in cells, you're reprogramming them. Eventually, if CRISPR-Cas9 is a treatment, do I inject it? Where do I inject? I mean, how does this translate into a real application potentially?
So I think we can break down the question into two potential applications.
Yeah.
First of all, I want to say that what we do in the lab is to treat the cells as a bulk of cells in which you transfect, so it means you basically transfer the protein for CRISPR-Cas9 into the cells with a guide RNA. The guide RNA is called as such because it goes specifically on a region of the genome, targeting precisely the genome. Of course, since it's always a stochastic event, we have to take into account that whatever CRISPR-Cas9 editing will always have some side effect, so there will be some other spot on the genome where it could potentially cut, even if it's not 100% the match of the guide RNA with the target genomic DNA. So this is what we do in the lab, and now we acquired at KAUST a very powerful instrument in which we can target single cell. So we can basically transfer Cas9 and the guide RNA in one cell and not to another one, and see whether the cells that has been corrected react differently and the one that is not doesn't, okay? So this is what we can do here nowadays. Instead, what you're asking more into the regenerative medicine applications, while I see, and this probably connects to Bruno's question, a lot of ethical concerns on the use of CRISPR-Cas9 on a living person, it's probably much more suitable the idea of performing regenerative medicine applications in vitro first, in which you derive, for example, skin that has been corrected by CRISPR-Cas9 in vitro. You can do all the possible assays on these cells that you derived in vitro, so to analyze whether there are mutations or side effects of the use of CRISPR-Cas9, and then use these cells that have been proved to be safe to be used for transplantation into patients. I think this is a more suitable option in the next decade, while the treatment of a patient with an injection of CRISPR-Cas9 in the bloodstream, I don't see it right now as something potentially available.
We got time for a few more questions. What else is out there? Yes.
Hello, my name is Vlad, and I have a couple of questions as well. So the first one relates to the fact that all three of the professors that we have here today work at levels of cell biology or above tissues and so on. These are systems with a very high number of variables that one can observe. Now my question is, you're targeting one, for example, one enzyme at a time that can be involved in multiple pathways, and you follow one of these pathways that you want to get a particular effect in. What can you say about the other pathway in which it may be involved? Like, for example, research can put a lot of effort into correcting a particular enzyme in one pathway, but can be very harmful in other pathways.
Yeah.
So this is the first question. And the second question is also related to the CRISPR-Cas system. So all these Cas and their derivatives that have been developed so far, it's somehow related to the first question. They are very good nucleases. At the end of the day, what they do, they deliver a cut. But you will still rely on other mechanisms like non-homologous and joining homologous recombination to come and kick in and do the job after the Cas delivers the cut. Now my question is, those other mechanisms that fix the cut, they are very complicated one, unlike Cas9 that is a simple enzyme. So what is the next step there?
Yeah.
Basically, for example, if you want to do, in a cancer cell, you want to deliver loss of function mutation, how do you know that you will block homologous recombination from fixing the mutation back for you?
So let me try and translate the biology question. I think they're roughly the same with different mechanisms. So you're saying this work that you're doing in a simplified system to control the variables, how do you start to ensure as it goes to patients that, or other types of treatment, how do you ensure that it's not affecting other pathways in the body because it's so complex? So who wants to take a shot at that?
Okay, I can start, I guess. So in terms of the enzymes, we're using them actually outside of the cell. So we actually decorate the cell from the outside, and then eventually, whatever's on the surface of the cell, it has a half-life, it's removed, and the cell is back to its normal state. And we also test that in the dishes. Like, we make sure that the cells are still, the stem cells still have the ability to give rise to all of the cells they're supposed to give rise to. So we can add differentiation agents and see that, for instance, a brain stem cell will still become an astrocyte, oligodendrocyte, and a neuron. But you're right, you know, if you are correcting or you are deleting a gene or you are doing something inside the cell, and you are then changing that protein or whatever it is, its role in not just the function that you're looking at, it could also have more diverse functions. And for those types of situations, we can look at omics technology, we can do proteomic studies and look and see, okay, if we're deleting or we're doing something to a protein of interest, is that affecting any other systems, any other signaling pathways within the cell?
So if I can add something perhaps to a higher level of variability, you touched a nerve when you asked this question, because when you do disease modeling, so you try to take patient cells, you reprogram them, and then you bring them back to the diseased tissue of these patients, you will observe an enormous amount of variability. This is because when you differentiate, differentiation is a stochastic event that you can try to trigger and drive with compounds in a dish, but you will never do it as well as nature does, okay? So the only way to try to mitigate the biological variability and the different persons' variability, because they have the different genetic background, is to try to increase as much as possible the numbers you are working with. So we never work with a control and a patient cell line, but we always increase the controls number to 4 to 5, and the patients to 7, in some case even 9 and 10, and per each patient we have at least 3 independent clones. So this is a way we use to approach the genetic and biological variability in these disease modeling approaches. For what concerns the Cas9, I'm afraid I forgot.
That's okay. I think it's along the same lines. I think part of what you're asking too is beyond the scope of what a university often does, right? At a certain point, they're collaborating with hospitals in Jeddah, for example. And then even beyond that, if something looked like viable therapy, you're going to do clinical trials at a scale and at numbers that make sure that some of these side effects are taken into account. So I think that's where, if they're as successful as they're trying to be, they're going to be collaborating at a scale where these can become suitable therapies. I got time for one more question. One more question. I see it there. Yes. Go ahead. Yes.
So I have a question to Dr. Jasmeen. So you talked about adding adhesive molecules to stem cells. Do you know if that affects the stem cell behavior? Because you have additional mechanical stresses. When it reaches its niche, it can also slide back out because now it has that option. So I don't know if you have, what do you have to say about it?
So in terms of the biology of stem cells, we can test that in the lab. And so if a stem cell is supposed to behave a specific way, and then we treat it with whatever it is that we want to treat it with, then we want to make sure that that stem cell is still behaving in the way it should be behaving afterwards. And we can also do that in vivo, so in animals. So once we treat them and we achieve what we want to achieve, so that they are, for instance, a blood stem cell is going to the bone marrow, then it's going to find its niche, and it's going to start to create those, all of the cells within the blood. So your red blood cells, your white blood cells, et cetera, platelets, et cetera. And you'll be able to know that those are coming from that human cell that we injected, because the mouse is a mouse system, and then anything that is human is going to come from that stem cell that we injected. So we know that the treatment that we're doing is actually not affecting its ability to give rise to all of the cells that are going to be functioning within that organism.
Cool. So on that note, you have plenty of time afterwards to come up and ask some additional questions. It seems like there's still more out there. I hope this was insightful in terms of showing you some of the work in biology that's happening at KAUST, and some of it is moving towards clinical applications. And also just, again, pulling back the curtain a little bit to show how modern biology works and show some of the tools that are being used here at KAUST and all over the world to develop some of these clinical therapies. So please join us next month during the Fall Enrichment Program. I believe we're going to be talking about marine biodiversity, some of the extreme things that happen in a marine environment. We hope you join us for that, and please thank the panelists for today's discussion.